know, then that might potentially allow you to have a quite kind of compact representation. So here's an example of that in terms of visual objects. Okay, so I don't know. Can you guys? Is this bright? Maybe I'd have to turn the lights off. This. Can you see there's like different animals here? I'm gonna hit the light.
I think actually.
Is that? Oh, yeah. Okay.
No, I'll just.
No, there you go. Okay, I'll turn it back on again.
Yeah.
Okay, so there's like a camel here, and a giraffe, and the kind of moose type thing here, and a pig.
What is that?
People like the moose type thing. Yeah.
Moose cow.
It's a moose cow. Yeah, I believe the technical term is a moose cow. Yeah. But it's some kind. They're different kinds of shapes. Okay. So if you were to say, describe to me in terms of low-level features, what is the shape of a giraffe? Okay. Well, just like you could describe a grapefruit and say, well, it's kind of, a large member of the citrus family with, internal segments arranged in such and such. You could describe the shape of a giraffe. You could, I could give you a kind of, 1,000 coordinates, and you could use those coordinates to build a kind of surface mesh of its shape or something like that, right? And in fact, when a computer draws a giraffe, that's exactly what it does, right? It has some kind of big list of coordinates. But you could also kind of say, well, it's kind of similar to, a horse with a longer neck, or it's, if you were to put all these different, what's a camel? A camel is kind of like a horse with a lumpy thing on its back, and this moose cow thing is kind of like a cow but with antlers growing out of its head. Okay, so if you represent these shapes in terms of their relations to other shapes that are on the same kind of level, then this can be a lower dimensional representation, a more compact representation. And so this kind of wavy, wobbly thing in the background is the kind of jargon term for that is a manifold, which basically just means a surface. So you've got this huge potential space. Say I was going to represent all of these shapes by like 1,000 points on a mesh, right? Then I have 1,000 dimensional space, right? But most of those possible spaces in that 1,000, most of those possible shapes in that 1,000 dimensional space don't look anything like animals, right? Most of them are just, like spiky, noise, cloudy things, okay? But a very, very tiny subset of them kind of look like animals, and all of those things that look like animals are actually in some ways quite similar to each other, right? Like they might all have 4 legs, and they might all have a neck kind of thing and a head over here, okay? So you can think of this kind of like lower dimensional space kind of living inside this high dimensional space, and there being a set of similarities on this low dimensional space. And so there's a theory, and this is just a theory, and this is actually something that I'm kind of sort of doing a study which sort of looks at this in a minute. There's a theory that this may be actually the way in which the brain represents the world. This is just a hypothesis. But if it does represent the world in those ways, then that could be a good kind of compact representation. And then if you ever do run into a moose cow type thing, you'll know what it is. Okay, where were we? Okay, so this is basically what the idea of similarity space is. This is one of the questions I asked some, and some of the answers I got made it clear that this wasn't really that clear so far. So, oops. So what is a similarity space? Similarity space is just literally the set of similarities between a bunch of items. Or it could equally be the set of dissimilarities if you just want to kind of, stick a minus sign in front of everything. So here's a very simple example. So suppose I got these 3 things. So we're going to look at just distance type similarities in this case, not this kind of set membership thing. So if I say, okay, I've got A, B, and C, and A is kind of close-ish to B and quite far from C, and B is kind of medium distance from C. So what does that end up looking like if I were to represent the full set of dissimilarities in this case? And I say, well, how far is everything from everything else? Okay, so I've got 3 items, so I end up with a 3x3 matrix. And this is just like, well, I guess you don't have these anymore, but back in the day, there used to be kind of road atlases, printed on paper, and they would often have a kind of grid of distances between different towns at the front of them. I guess they don't exist anymore. But anyway, so if you were to plot it, if you were to write out a grid of how far everything was from everything else, okay, everything is zero distance from itself, right? Everything is perfectly similar to itself. So in a dissimilarity matrix, so the line of things with themselves is always this leading diagonal here. So like A, the distance from A to A, distance from B to B, distance from C to C. Okay, so in a dissimilarity matrix, they're all zeros. In a similarity matrix, there will be ones for like the meaning perfect similarity. Okay, so you know, A is kind of close to B, so the distance is say 1 here. You know, A is really quite far from C, so it's distance of 5. So the similarity space is just this set of distances. So the two things to notice about this is that everything is perfectly similar to itself, and also it's symmetrical in this particular case. I mean, the Luxembourg and France example was asymmetrical, but if we're just looking at distances or other symmetrical measures, then your similarity matrix will be the same as if you flip it. So symmetrical measures. So that's all that similarity space means. But it is something that people have actually looked at a lot over the history of cognitive science, and also people look at it a lot in computer science and in machine learning as well. So we were looking a little bit about, you know, what is a classifier and how does a classifier generalize? And essentially, the way a classifier generalizes is probably a little bit like the way we generalize. Is, you know, encounter something new, and it says how similar is it to stuff that I already know. If it's highly similar to stuff that I've previously assigned to class A, then I'm going to assign this thing to class A as well. If it's highly similar to stuff that I've assigned to class B, I'm going to assign this thing to class B as well. And you know, clearly in this case, you have to say, well, similar in what sense? So here it has a particular algorithm. It says, well, okay, I'm going to measure similarity by, you know, looking at the correlation of the coordinates or something like that. So, so people have looked at this a lot in, you know, over the last more than 50 years. So one approach that people have taken, and this was actually pioneered by Roger Shepard, who's that guy I mentioned before, is multidimensional scaling, which is a very useful method with a very horrible name, in my opinion. I mean, you know, if you just look at the name multidimensional scaling, that really doesn't tell you anything about what it actually does. What it does is it takes a set of similarities and tries to find a low-dimensional representation of them. I'm going to show you a picture of that in a minute. And this is actually also a very useful approach that we're also going to encounter in fMRI papers, and also pretty much in any type of paper that looks at similarities, actually. So let me show you an example of that. Actually, I'll skip these other ones for now. Okay. So this is a nice example of multidimensional scaling from Kriegerskotte, who we've been mentioning quite a lot, and also Rusbey-Kiani. So this is I'm going to hit the lights again. Can you guys more or less sort of see what you might not be able to see what all the pictures are, but can you see that there's pictures of stuff? Yeah. Okay. So this is actually 96 different visual objects. Okay. And there's let me tell you a little bit about what this image shows. So this takes took a bunch. What Kriegerskotte and Kiani and colleagues did was they took a bunch of different pictures of different types of classes. So some of them are kind of faces, some of them are body parts, things like hands, arms, stuff like that. Some of them are entire human figures. Some of them are pictures of animals like elephants, frog, monkey. Some of them are faces of different animals, so like a lion face and a baboon face here. Some of them are natural objects like fruit. Some of them are artificial man-made objects like a key or a chair. Some of them are scenes like a picture of a forest with trees in it. Okay, so there's all these different types of objects, and they showed them to people, and they showed the exact same pictures to monkeys. And in the people, they recorded multivoxel patterns of activation in ventral temporal cortex, so this kind of part of the brain, kind of near the bottom of your brain, which is very sensitive to objects. This is the same area that the Huxley paper looked at. Lots and lots. Anyone who looks at object recognition looks at ventral temporal cortex because that's where a lot of object-sensitive brain areas are. And looked at the multivoxel activation patterns, and then looked at the pattern similarity between all those multivoxel activation patterns. In monkeys, this is the part that Rusberg-Chiani did, instead of doing human fMRI study, they showed it to monkeys, and they did recordings from individual neurons, but they were recordings from lots and lots of individual neurons. I think about 300 or so, actually. Actually, it was sequentially. If you had a sufficiently nice electrode array, you could even record from 300 neurons at the same time. Well, that's kind of hard to do just for technical reasons. But what Rusberg-Chiani did was he actually just recorded individually sequentially from all these 300 or so neurons, and then you get basically a kind of activation vector of activation across that population of 300 or so neurons, and then looked at the similarities of all of those activation, single neuron activation, well, neurophysiologically recorded activations, again just using basically correlation as a measure. And so then what you have is, you've got 96 different pictures, and every picture has an activation pattern associated with it. So if you look at the similarity of each picture to each other picture, in terms of their neural activations, you've got a big 96 by 96 thing, okay, which is thousands and thousands and thousands of similarity values. And so this is a very, very high dimensional space. You've got thousands and thousands and thousands of dimensions. And if you say, okay, well, what does that look like? Right? Well, it's kind of hard to draw a picture or even get any sense of the structure, intuitive sense of the structure of a space with thousands and thousands of dimensions. But we can draw things in 2 dimensions, and you can kind of sort of draw them in 3 dimensions if you do them with a computer and spin it around or something. But 2 dimensions is much easier to visualize. So what would be nice would be a way of taking very high dimensional space and finding a way of representing it in low dimensions. And that's what multidimensional scaling does. So here's actually an example. This is a very easy example, right? Here's an example of a 3-dimensional space, right? Because I've got a 3 by 3 grid here, represented in 2 dimensions. Okay? So I've got the distance between 3 cities and 3 other cities, and I can draw it in this 2D map here of this thing's quite far from that, and this. Now this is actually let me put I can put the light back on it. Okay. This is actually a perfect representation of that. I know it is because, this is actually just describing the distances here. Okay, so in this unusual case, you can actually reduce something from. I mean, it's not much of a dimension reduction, right? Going from like 3 to 2, but you can have something in a lower dimensional. Is it clear how this is like a lower dimensional thing? Because this is just a 2D picture, and this is like three cities, okay? Okay, what if I had 1,000 cities, right? Now, if your 1,000 cities were actually literal cities on a map, and you just plotted, literally measured all the distances between them, then again, you could have a perfect representation on 2D. It would be your map that you started off with in the first place of the distances between all those cities, okay? But that's obviously kind of like the best case scenario, right? In general, this set of distances, if it wasn't actually made in the first place by measuring things from a lower dimensional 2D representation, it has no obligation whatsoever to allow you to represent it in some kind of nice 2D way without any distortion or any loss. Okay, so you're always going to get some, in almost all cases, you're going to get some distortion or some loss. But if you but you can say, well, what would be the way of doing it that would give me the least amount of distortion? And so you can imagine, and basically this is what this algorithm of multidimensional scaling does. It says, try and find a way of representing things in low dimensional space that kind of distorts this as little as possible. Okay, so you've got your true set of measurements, which is, in this particular case, 96 by 96, so thousands and thousands of dimensions. And then you've got a huge, huge reduction in dimensionality going down to 2D. But in many cases, not always, but in many cases, the multidimensional scaling can actually do a pretty okay job of capturing at least the broad main aspects of the structure without, clearly some information is lost. Anytime you do any kind of reduction of dimensionality, some stuff is going to get lost. But maybe, hopefully, if you're lucky, the crucial things won't get lost. So what did they find? Well, it turns out, and this might be easier to see here in these little colored dots, actually. So what the colored dots show is that the faces are kind of colored as I think is kind of red, the bodies as a kind of shade of purple, natural objects as a kind of darker blue, artificial objects as lighter blue. And if you look at, say, the monkey IT here, you can actually, well, maybe at least if you're near the front, you can see, and I'm going to put this slide up on Piazza so you can look at, the actual original paper. Oh no, the original paper is not up in Piazza right now, but you can put it up there. Or you can find it on Google Scholar or something. There is a lot of structure. Whatever this dimension reduction has done, it does seem to have preserved quite a lot of structure. Because look, all the faces are kind of bunching together here. A lot of the body parts are kind of bunching together here. A lot of the natural objects are kind of bunching together here. A lot of the artificial objects, or seams, are bunching together here. So in a very kind of, so the coarsest possible split of this is that everything on this side is kind of natural. Right? Oh, sorry, everything on this side is kind of animate. It's stuff that moves, right? It's basically animals and faces and human faces and animal bodies and human bodies. Okay? And everything on this side is inanimate, stuff that doesn't move, objects, whether they be natural or artificial, vegetables, keys, road signs. None of those things will move on their own. And it actually turns out that this split between animate and inanimate seems to be something that's quite fundamental to what the brain does. In fact, this was actually first discovered, I think in the 60s, by people like Warrington and Chalice. There's certain types of, certain patients occasionally who with brain damage to particular areas, typically areas in ventral temporal cortex, suddenly have great difficulty recognizing or naming entire categories of objects. So you can show somebody, anything that's actually animate, and they might be like, I can't say what it is. But you show someone something that's visually just as complicated, or visually, has many overlapping features, but which is inanimate, and they're fine. They'll say, oh yeah, it's a road sign, it's a key, it's a banana, it's an umbrella. And so this is actually, this is quite surprising, because this is a very kind of high-level split. If you were to say, give me some low-level visual feature that all of the animate things have and all of the inanimate things have, there isn't really one. I mean, a lot of the animate things have eyes, right? But not all of them, right? Like the hands and the body parts, they don't have eyes, right? Or like the picture of someone's ear, right? That's just over here. Okay, so it's not something as simple as just does it have eyes or not. There's some kind of quite high-level conceptual split between animate and inanimate, and then there's also kind of subdomains within that. So all the faces are together, things like that. And then if you look at the similarity space in human IT cortex, it has an incredibly similar structure. So all the faces are over here, all the body parts are over here, all of the natural objects are up here, all of the kind of visual scenes are down here. And these kind of color-coded representations sort of captures that too. So it very much looks as though human inferotemporal cortex, and inferotemporal, ventral temporal, basically just other ways of saying the same thing. So IT stands for inferotemporal. Human inferotemporal cortex, monkey inferotemporal cortex seem to be carving up the world in terms of similarities in very, very similar ways. So this means you can actually make a real comparison between human and monkey representations of the world. But if you think about it, what's actually being compared here, that's actually kind of amazing, because they're two completely different types of data. Okay? So this, this is fMRI data. This is recorded from a different species, but it's totally recorded in a completely different way. It's slicing the brain up into voxels, making this indirect measurement that pulls over literally millions of neurons at once, and is actually just measuring blood oxygenation. Is recorded from, say, 1,000 voxels. This thing, as well as obviously being from the different species of monkeys, is recorded by sticking an electrode into a brain and recording changes in voltage at the end of a little tiny electrode, and doing that lots and lots of times. And over a different number of neurons. So this might be over, say, 300 neurons, and this is over, literally millions, some big pool of signal over literally tens, hundreds of millions of neurons. And yet, despite these completely different types of data, something very, very similar emerges. And if you were to look at, if you were to take just the raw data and say, can I compare this? Then it seems almost impossible even to compare them. If you say, please tell me how similar, what's the relation between this bunch of 300 single neuron recordings that I have and this bunch of activation from 1,000 voxels from a human being? You'd say, well, I don't know, that's like, it's like chalk and cheese. They're just different things. Right? It's not even clear how you would even compare them. Okay? But you can certainly compare activation across 1,000 voxels when someone's seeing, a pair of pliers to activation against the same 1,000 voxels when someone's seeing a chair, right, or a monkey face, or an alligator. Okay, right? You can do that. You're comparing the same kind of stuff with the same kind of stuff. And similarly, in the monkey data, you're comparing activation across a bunch of different neurophysiological recordings. It's the same stuff with the same stuff. And then, having done this kind of within recording type comparison, now suddenly they're both in the same. They're both in the same structure. They're both a bunch of similarities. Okay, you've got a 96 by 96 matrix of similarities from the human, and you've got another 96 by 96 matrix of similarities from the monkeys, and those you can compare. Okay? So this was kind of one of the questions, and those of you who haven't sent it in, which is totally fine. This is basically the answer to that question. But please try and give a compact, a lower dimensional representation of it. So you've got. This is how you can take things that are very, very different and kind of make them, if you like, speak the same language and then compare them to each other. So I'm not saying that's the only way of doing it, but similarity space is a very powerful way of doing that. And these guys, I love this paper so much. These guys were the first people to do that. I think it was really the first people to do it just in general. And certainly the first people to do it comparing across different species. Before that, it was really very, very difficult to ask the question, what does a monkey's representation of the world look like compared to a human being's representation of the world? And in order to understand, in order to understand the brain, that's kind of what we want to do, right? We want to try and find what's common across, what's common across brains in general, right? It's amazing how much a monkey brain is like a human brain, especially if you look just at kind of, lower level visual things. I mean, once you get to stuff like language, then, the monkeys don't have it. But if you look in terms of being able to deal with a noisy, complicated visual world, monkeys can do that probably better than we can of anything. Okay? So there ought to be a lot in common between our representations of the world and monkeys' representations of the world. It's very hard to know how you would be able to ask that, and this is a way of doing that. So, so in the next, so on Thursday we're going to look a little bit more at not just relating between different species, but say finding commonalities between different people. And we're going to kind of look at some of the kinds of things you can do with neural similarities and psychological similarities. So Thanks very much. If anything is unclear, which I'm sure some stuff is, please, email me or post it on Gas or something like that.
Well, I got here. No, I had monkey problems, and I was the only one qualified to fix the monkey problem. Having problems. Yeah, there's just basically like after you record, the connective tissue starts to grow back over the dura because of all the piercings that are within it. So what can happen after that is you'll get swelling like of the dura and the connective tissue, and there's like an increase of blood clotting, et cetera, et cetera, et cetera. Alex doesn't know how to take care of it. I was Joker. Because we just recently stopped recording from him, and it's a relatively common fault.
You have to just scrape it.
That part was not really necessary.
Very, very carefully. Because the thing is like when you're scraping, you don't want to like, there'll be like a little divot and you can't go under the divot and lift it off. You have to like very, very carefully like erase it. But it was like, I was there, I was just signing up now.
I'm feeling really good about it lately. Practice so much. But now I'm all of a sudden getting really nervous.
Yeah, like twice a week.
Getting in the room. Yeah, but when you're cleaning, I want to make a good
So it's like
impression.
growing, that there is like all of a sudden, like does it just happen all of a sudden? It just happens all of a sudden. It happens like you cleaned him Friday, it's Tuesday, and like you can kind of see the connective tissue is like growing back.
It's like 180 slides.
But it's not
But
really
the presentation's only 60, and then the rest are 100
just in case it works.
ask me questions about other things that I couldn't have data for. But
Yeah, I mean this was like.
yeah, I want to practice it because you can tell like when you sit here it cuts off the film. So I want to scooch things up on PowerPoint to make sure
But in
that.
this case, it's not
Okay.
Thanks for that, by the way. No, no problem.
Is it just open to like?